To get started, I'll give you a little bit of an overview. So the next 4 labs are associated with the lecture module on sex and gender. In society today, understanding the biology behind sex and gender is important in order to understand why we see so much diversity in people. But beyond the basic biology determining sex and gender is the societal view of this diversity and why we as people have so much difficulty embracing this diversity. So in this series of labs, you will use an evolutionary perspective, delving into why our society focuses so much attention to sex and gender, why we see so much diversity, how our societal norms come about, and finally, why it's so difficult to fight against these deeply entrenched societal norms. So one paradigm associated with human behavior follows the premise that people are different from other organisms and should be viewed in isolation because rules and principles associated with other organisms don't apply to people. In contrast, a second paradigm views all organisms, including people, as a product of evolution, and as such, people are subject to the same rules and principles as other organisms. Thus, by evaluating other organisms and the overlying evolutionary principles, we can gain a greater understanding of why our society functions the way it does. So, in this series of labs, we'll use the second paradigm that people, like other organisms, are products of evolution. And by studying other organisms, we can get greater insights into ourselves. So in these labs, we'll look at fungus reproduction. We'll look at plant reproduction and the nature of mating systems seen in animals, and relate specific aspects of these organisms to overlying evolutionary principles, linking what we study about these organisms to people. So let's see. So to start, our first lab delves into the question, why do we have sex? So think about it for a second, seriously. Why do we have sex? Why is our society so over-occupied with sex? You turn on the TV or listen to the radio, and sex is most likely what you see or hear. Why is the concept of love so important, and why do so many people spend so much time and resources on finding it? So when asked, most students say the reason we have sex is to reproduce. Nothing could be farther from the truth. Think about it. Sex is hard to do. You have to find a compatible mate, you have to woo the mate, spend a lot of time and energy finding the mate. Think about how much time and money you spend on finding a mate. Now imagine if you could just grab some cells, throw them on the ground, and have them develop into another person. That would be so easy, and from an evolutionary perspective, right, it's possible. It could easily evolve. So why do we have sex? Okay. Why do we go through all of that time and effort? So other organisms use asexual means to reproduce. But even if they can reproduce asexually, they still spend a lot of time and energy on sexual reproduction. So we're not unique in sex, and sex is pretty much universal. If you think about bacteria, even bacteria have the equivalent of sex. So why do we have sex? Right? Now, if you think about it, sex is the union of 2 genetically different organisms. Therefore, the simplest explanation is that we have sex to make genetically diverse offspring. If that's the case, then we should see mechanisms at the cellular scale in almost all organisms, including people, that help to create genetic diversity. In this lab, we're going to look at a process that occurs in basically all multicellular organisms, including people, and that process is called crossing over during a second process called meiosis. So to examine crossing over, I've chosen to use a fungus called Sordaria formicola. So we'll take this lid off. Here is a plate of Sordaria formicola. If you look right here, Sordaria formicola have different strains. These different strains are analogous to our sexes, so we have plus strains and minus strains, right? So here we have a strain. This is all both types of a single strain called wild type. Over here we have another strain called tan. Okay, those strains then are determined based upon the spores and the spore color that are produced. So I put a tan strain here and a tan strain here, a wild type strain here and a wild type strain here. Then we have little filamentous structures called hyphae. Those hyphae have grown out from our initial inoculation point. And you can see right here in the connections between these different strains, we have sexual reproduction. So all of these teeny tiny dots are basically the products of sexual reproduction in this fungus. And what we're going to do in this lab is that I'm going to take a scalpel, I'm going to scrape off some of these little teeny tiny reproductive structures, which we'll talk about in a minute, get those, look inside those, and then we'll look at the process of meiosis by examining this Sordaria formicola or fungus. Okay. So the techniques we'll be using here then to analyze these fungi will be that I will be making a wet mount. So here I have a different type of microscope than what we looked at before. This is a dissecting microscope. We can see here no objectives down below here. Okay. My plate of Sordaria formicola that we looked at a few minutes ago now is on the dissecting scope. I have it hooked up to a camera. The camera gives us a live feed of the things on the plate. Okay, so now we'll move to a view from the microscope. Before we go to the microscope, though, I want to just quickly mention how a fungus reproduces. So if you think about a fungus, you go out in your yard, you see a mushroom, right? That mushroom is the product of sexual reproduction in a fungus. So here we have all these little teeny tiny dots. These little teeny tiny dots are the equivalent of a mushroom. So what happens is we have one strain here of all these filamentous structures called hyphae. We have another strain over here. Our hyphae then come together and they get together. Okay. Where these hyphae get together, then hyphae are a fungus. The main state of a fungus is called haploid, which means it has one set of chromosomes. In people, our main state is diploid, meaning that we have two sets of chromosomes. So our life cycle, we are diploid, we go through meiosis, we make sperm and eggs. Our sperm and eggs get together and make a diploid adult. In fungus, their predominant stage is haploid, being these hyphae that occur everywhere, big giant mats of hyphae all over the ground. Then when one strain meets the other strain, those two strains get together and make a mushroom. That mushroom then has a diploid stage. When it becomes diploid, it goes through meiosis, making what are called haploid spores. The teeny tiny haploid spores then are released from the mushroom, go fly out, and land on the ground somewhere and then grow into hyphae. Okay, so that's what's going on here, is that we had spores here, the hyphae then grew out, those hyphae grew out into mushrooms. All these little black dots in here are mushrooms, and we're going to look at the actual spores inside these mushrooms. The interesting thing about these spores is that these spores are colored. So our tan strain makes tan spores. Our wild type strain makes black spores. So we're eventually going to move to a compound microscope and look at the distribution of tan and black spores in these mushrooms. So here's a view through our dissecting scope of these little teeny tiny mushroom, or the equivalent of mushrooms, in the Sordaria. I'm going to go ahead and zoom in. Dissecting scopes, then you can do things too. And so I'm going to zoom in. Then I'm going to focus. We can see these individual mushrooms. Inside these mushrooms then are spores. So basically spores are the reproductive structures of fungi. these little teeny tiny mushroom-like things, which are called parathecia. These mushroom-like things are actually little cannons, where you can see they're shaped like a cannon. The top of them explode off and they launch mushrooms way out into the air when they're all mature and ready to go in and reproduce. You can see all of the fine hair-like structures in there. Those are the hyphae. You don't necessarily have to remember anything about the hyphae. You don't have to remember the word parathecium or anything. What you really need to understand is that these are mushrooms. Inside these mushrooms are spores. And we will be examining the spores in a few minutes when we make a wet mount and look at them under a compound scope. All right, the next step is to actually make the wet mount. So to make the wet mount, I'm going to take a water bottle and we'll put a drop of water on the slide itself. Okay, so I have a little bit of water on the microscope slide. Move the water bottle. Now I have a Bunsen burner going. Anytime you work with bacteria or with microorganisms, you want to make sure you use a sterile technique. So in this case, the fungus doesn't get infected by other fungi or bacteria. So I'm going to flame my loop. Now the nice thing about dissecting scopes is then I can use the dissecting scope to watch and see exactly what's going on in here. Okay, so I'm going to use this dissecting scope. I'm just going to scrape carefully off some of these parathecia. I don't want to get any augers of the technique, right? I'm kind of taking it back and scraping it like I'm shaving these little guys right off of there. I'm going to take all of these guys and I'm going to put them into the water. Reflame my scalpel so that I don't have any fungus on the scalpel. Put the lid back on the plate. Then I'm going to grab a cover slip. We'll put a cover slip gently on all of these little microorganisms. Now in this case, the final step that we want to do is that we want to take a chem wipe and we want to actually explode those parathecia so we get all of the spores out of the parathecia. So basically I'm going to take this. We'll just put the chem wipe on top and gently press down to explode all of those little parathecia, little mushrooms. So here's the view through the microscope of actually picking these up with the scalpel. So you can see the scalpel there. And so we go down and we just shave off. We grab these little guys. And if we look very carefully, I can focus in on that. So I can see all the little teeny tiny parathecia on the end of that scalpel blade. So here are all of our little mushroom guys in there. So each one of those little black dots is one of the mushrooms that we have in there. And then I basically push down on the cover slip to launch out the spores. So right in the center I have one that's really good. We're going to zoom in on that one underneath this dissecting scope. And I'm going to go all the way in and I'm going to focus, because as you zoom you actually have to change the focus. And there we go, that's really nice. We're going to go ahead and look at this underneath the compound scope here in a couple minutes. That's as close as we can get on that guy. But we can see that guy really well. We can see how I pushed on it, I exploded it, and then out of that parathecium or mushroom, then we can see how we have all of those chains of spores stuck together. So you have kind of a little rosette of chains of spores all sticking out on that guy. Now we're going to look at these guys underneath a compound scope, get up under a lot higher magnification to be able to do the next step in the estimate. What we have here is a compound microscope. We're under the 10x objective for a total of 100x magnification. The way that these fungi reproduce is that they go through a round of meiosis, then they go through a round of mitosis to make spores. So in the round of meiosis, if you think about people, one round of meiosis in people, right? We start with one diploid cell and we make 4 haploid sperm. Okay, so these guys, the meiosis is exactly the same, where they go through one round of meiosis and make 4 haploid spores. Then they go a round of mitosis to make 8 total spores. So we're looking at these sacs of 8 spores, and in here you can see a lot of sacs of 8 spores. Now it takes time for the color to get into the spores, so we're really looking for mature spores with 8 sacs that are 4 of them tan and 4 of them black. So if we focus in here on this group right in the center there, we can see that group in the center has 4 tan and 4 black spores. So see, that's a 4 and 4. So in your lab report, then you would tally that one as 4 tan, 4 black. Okay. Unfortunately, in this particular one, we only have one really good one, 4 and 4. And so what I'm going to do is I'm going to look around, and then we'll spot groups of 8 where we can get a nice group of 4 and 4. So that one was looking good, except it only has 6. There's one with only 2. So not great. Now we're going to search around some more. And so I'm going to click this off, find some, then we'll focus in as we find them. Here's a rosette where a lot of them have their color, so we'll increase magnification. We'll kind of move over on these guys here. There we go. Okay, and only ones that we can fully get a count of, we want to count. So there's one good one that we can see there. A lot of these don't fully have their color yet. There's 2 that we can see 8 cells in. Okay, but if you can recognize any and get them fully counted, then you want to record those individuals in there. Okay, and I'm not quite able to get full. There's a 4, 4. All right, I'll stop it and go else. So here's a good one. You can see that it's not fully gotten its color yet. We can see kind of a 2, 2, 2, 2 arrangement there. Okay, here's another one. Let's see what we have here. You have to be able to get all 8 spores, and they have to be fully developed. But there's one right there. So you can tally that one. Some of these others haven't fully gotten their color yet. There, it looks like maybe one at the upper. Have those 4 black spores. No, can't quite get it. No, not that one. Here's a nice one of 8 intact spores. Dead center, we can see that one pretty clearly. That's nice. Okay, in the center of this one, there's a nice one. We can see clearly, ooh, a really nice one. Okay, we have one here, if I can get it in the center. There we go, you can see that one nicely. Right there. Good 8 spores. Here's another nice one. I'm focusing on that guy. Okay, the one below it doesn't quite look mature, but that one is mature with very good color. Okay. Okay, there's another one. This one we can see there, dead center. Nice group of 8. Here's actually another one. Right there, kind of on the left side, you can see that group of 8. That distinct pattern. The others look like they're too jumbled to see. Okay, we have several here. Some of them aren't totally mature. Only one there we can really see well. The other's not as well. Got one there. There's a nice one. Okay, looks like several here with this one that we can see. If you look around, you can get quite a few of them. So any of those there that you can make out, you can find, you can use. There's 2 of them right in the center there that you can use. This one only has 7 spores, but it's looking pretty good. The one next to it is good. Another nice one there, and you can see with this scope, once you figure out what you're looking for, we don't have to zoom in on higher magnification every time. We know exactly what we're looking for. We can go a lot faster under lower magnification rather than higher magnification. Okay, several good ones in this image. Put those right in the center. We can focus in and out. Sometimes further away you can kind of see those colors better than close up as well, but even those 3 right there on top, you can get all 3 of those. The one to the left over there.
Okay, and a few of those guys as well. Alright, looking pretty good.